This is a little bad topic at the end of the day because it's a little difficult to grasp, and the problem is you require a lot of attention all the time to understand what I'm going to talk about. So please excuse me for boring you with this topic at the end of the day, but just bear with me. I'll go slowly, and this is a totally an illustrative lecture, very few written slides so that you can grasp what I'm going to tell you. Now, to talk about microcirculation, what do we try to understand? There are certain structural components which define the basic anatomical microvascular module. You have the artery at one end, which divides into arteriole. Then it goes into capillaries, and these capillaries unite to form venules and veins. So there is a continuous channel and a continuous communication onto the arterial side and the venous side. Now, what is the difference between an artery or an arteriole and a venule? It is the tunica media coat which differentiates between the two. So if you look at them, you'll find that arteriole has a thicker tunica media, while a venule has a very thin tunica media coat. That's how we differentiate between venules and arterioles. Looking at the arterial structure, the outermost coating of the artery is made up of collagen. When we strip off the collagen, you'll find the tunica media consists of elastic tissues and muscle cells. And we take out the media, then it is the intimal or a single endothelial layer which lies there. So these are the three layers. And I told you it is the tunica media which is made up of elastic fibers which constitute or differs the arteriole from a venule. So why do we need an arterial structure like this? Because this forms a pressure reservoir which provides a continuous flow and pressure for the blood to be driven to smaller capillaries. Otherwise, if we have low pressure, once the pressure drops, there will be no flow to the capillaries or to the tissues. So the capillaries are responsible for tissue blood flow. Now, when this reservoir or when the heart contracts, this reservoir expands, and when the heart relaxes, this reservoir passively recoils. So there is an intermittent expansion and recoil which gives you a pulsatile flow to the tissues. That's why we say that pulsatile flow is important for basic physiology rather than a continuous flow which you get in a cardiopulmonary bypass machine. Now let's see the arteriolar structure. Now when you see the arteriolar structure, you'll find that the arteries have concentric smooth muscles on top of it. They are circular in shape, and these are the ones which can constrict the artery further, the arteriolar further, and therefore these muscles are present all around the circumference of the arterioles. There are elastic fibers and there is endothelium. Now what does these arterioles do? These arterioles provide major resistance to the blood flow. So these are the resistance vessels, and that is how we calculate our systemic vascular resistance. These are the ones which are giving us those resistance. Now this tonicity of arterioles is actually governed by three factors. The first factor is the neural control. The second one is the humoral control, or the hormones which act on it, like the norepinephrine. And the third one is a local control. So all these things mediate together or work in tandem to give you the tonicity of the arteriolar system. Now imagine if the arterioles were not there and there was no resistance, then what will happen? It will be like a patient in total septic shock. There will be no blood pressure to come up. So if there is complete vasodilatation or no resistance at all, these patients would fall flat. Now, how does the nerves control the tonicity? All these arterioles have a neural sheath over them, and this are basically governed by alpha 1 and alpha 2 post-junctional receptors, and they are the ones which are responsible for sympathetic vasoconstriction. What about the distribution? You'll find small arteries and arterioles are governed mainly by nerves, while as the meta-arterioles at the microcirculatory level, the prescapillary sphincter and muscular venules are governed by the local control or the local environment around the capillaries. So this local autoregulation is called as vasomotion. Now, why is this vasomotion so important to us, which provides a local control, regulatory control over capillary circulation? Now look at this. You have the capillaries here. This is the arterial one. This is a meta-arteriole. There is a capillary network, but there is a small flyover. That is a thoroughfare channel which connects the arteries directly to the veins. And when we talk about AV shunting in sepsis, that the arteries are, the blood is getting shunted off, it is through these channels that the blood gets shunted off. And they don't have any precapillary sphincters here. They are only present in the capillary network which is present there. So the blood can go through these thoroughfare channels at times. And what does it do? These capillary sphincters, precapillary sphincters, the meta-arterioles, they regulate the by regulating the smooth muscle contraction. They give intermittent flow to the capillary, and this is what is defined as vasomotion. Now, what's the use of this? Why should we have thoroughfare channels, and why should we have this nutritional capillary network? So to understand this, we'll go to the next slide. But remember, one of the local factors which causes the relaxation of precapillary sphincters is the decrease in PO2 or hypoxia. And that will lead to sudden release of these precapillary sphincters so that the blood can flow to tissues unhindered without any sort of resistance to it. So that's a protective mechanism which takes into action. Now, why do we have these thoroughfare channels and nutritional capillary network? So consider this is your skin surface. You have a small artery, and this artery divides into a network of nutritional capillaries which supplies the skin. It's a large network, and below that there is a continuation of the artery and it terminates into the vein. This is a thoroughfare channel, and this is known as a thermoregulatory AV shunt. So those thoroughfare channels which I described are called the thermoregulatory AV shunts. How do they work? Now let's consider a condition called hypothermia. Now the hypothermia, when it happens, you'll find that the AV shunting is on. That means there is peripheral vasoconstriction so that less blood is flowing through the nutritional capillaries. Most of it is being shunted off by the thoroughfare channels. And this is in protection, because the protection is that against further blood heat loss. The more the blood flows through the nutritional capillaries, the more the heat loss will be through the skin to the environment. So it's a protective mechanism that there is constriction of nutritional capillaries, and most of the blood will be shunted off through the AV regulatory shunts through the thoroughfares onto the venous side. And together with that, you will also have piloerection, which is normally seen, that the hairs will be standing up. So this is a response to hypothermia where you have vasoconstriction. What happens in hyperthermia? Just the opposite. Opposite means that in hyperthermia, body wants to get rid of the heat which has, and it has to enhance the heat loss to the environment. So the auto shunting will be off, and a lot of blood will flow through the nutritional capillaries. That's why you have the hands which will be red. They'll be flared up because the body is trying to lose heat, and that is how the heat loss is governed through the skin by this kind of an autoregulatory AV shunting on and AV shunting off. Along with that, to cool the skin down, you will have sweating. So as you sweat and as the convection currents flows, the body surface cools down. So this is an example how vasomotion helps you to regulate temperature or body temperature. Having understood this, Let us just look at the body fluid compartments. Now, if the total body water is about 42 liters, then you will find 2/3 of it is the intracellular fluid and 1/3 of it is the extracellular fluid, and both these fluid compartments are separated by a cell membrane. Now, if you want to divide the extracellular fluid, which we will normally call as, we will find that the plasma is about 3.5 liters, you have a capillary wall, and the interstitial fluid is about 10.5 liters, which is quite high. So for the fluid hemostasis, this is the microcirculatory environment which we need to talk about. That is the interstitial fluid, the plasma volume. So this is what we will be having to deal with to understand the microcirculation properly. So how does the fluid hemostasis takes place? There is a partition between the interstitium and blood vessels, and that's the plasma. There has to be one. There is a cell and the interstitium which are separated together, and there is interstitium and lymphatic vessels. So these are the three divisions of the compartments which we need to understand. The plasma and interstitial fluid always have a similar composition. The intracellular fluid always remains constant. So it always remains constant, it doesn't change. So if there is a change in intravascular volume, then you will find it has a direct influence onto the intracellular as well as the interstitial water because of the hormones like ADH which are secreted. But what happens to the cell? In hypervolemia, you will find there is higher interstitial pressures, and that can lead to cellular swellings. But in hypovolemia, the interstitial pressures are likely to fall down, and that will lead to cell shrinkage. There is another condition where cells can likely to swell. Specifically, when the plasma membrane is injured, then everything goes inside the cell and swells it up. Now, the most important part is the role of lymphatics. These lymphatics are present, and these lymphatics play a major role in fluid hemostasis. How do they work? Now look at this. This is the interstitium here which you see, and this is your lymphatic vessel wall. The lymphatic vessel wall has larger interendothelial gaps. So there are endothelial gaps, and the water which goes against the pressure gradient enters into the lymphatics. So this is dependent upon the pressure gradient. So once the water enters the lymphatics, the pressure inside the lymphatics here would build up. And once the pressure inside the lymphatic builds up, all the endothelial pores are closed. And that is how the lymphatics work to remove the excess of water from the interstitium and put it back into circulation. So whatever comes to the interstitium has to be removed by lymphatics in this manner. Right? Now if there is edema, if there is more of edema or the interstitial pressures are high, then what does it do? The capacity of lymphatics to drain that interstitial fluid cannot be enhanced. It will remain the same unless there is some way we can dilate the lymphatics, which is not possible. So the only way the interstitial fluid or the excess of interstitial fluid can be reduced is by decreasing your hydrostatic pressures or by making sure that the cell injury stops. So whenever the lymphatic starts draining more than what is coming into the interstitial cell, the patient's condition is likely to improve. Now, considering the flow, the pressures and the volumes, the capillaries here, if you look at, you will find that the velocity of flow is the minimum in the capillaries, whereas if you take the cross-sectional area, you will find that the cross-sectional area is the maximum in the capillary network. And if you take the mean pressure, there is a gradual decline from the aorta to the arteries to arterioles to capillaries, and it is least in the venules. That is how the flow governs, because according to the pressure gradient which falls gradually from the arteries to the capillaries and to the veins. Now look at the endothelium layer. When you look at the endothelium layer, we considered or believe that the endothelial layer inside the vascular lumen is completely smooth. until we were shown that it is not so. This endothelial cell lining inside the lumen has something which is called as glycocalyx. These are hair-like structures which grow inside the lumen of the endothelial capillaries. And they actually look like this. They are gel, sometimes they are gel surfaced, or they are hair-like structures, or it's also called an adsorbent layer or a endothelial glycocalyx layer. We could not find them earlier because we didn't have better staining methods. Whenever they tried to stain, they used to destroy the endothelial glycocalyx, and it was not visible during staining methods. This glycocalyx, which is present, is actually an active interface between blood, which is flowing into the capillary, and the capillary wall. Just remember this. This is the most important part of the vasculature or the endothelial vasculature, which we are going to talk about. Now if we talk about the fluid body compartments, you will find that this glycocalyx layer also has some fluid which is arranged. It is not participating in circulation, but it does have about 2% of the plasma volume which is stagnant into the glycocalyx. It remains there. So this is how the fluid compartments have now changed, that 2% of the plasma volume is contained within the glycocalyx, which forms a gel kind of a surface over the endothelial surface layer. Now how do these glycocalyx help in transfer of different fluids or different ions or different particles or different molecules across the endothelium outside into the interstitial space? So just have a look at this particular diagram. Now here if you see, we have the endothelial glycocalyx here. This is the endothelial lining, and this is your basement membrane, and this one, white one, is the interstitial space, and this is the lumen of the capillary wall. So there are different types of adhesive molecules. One of the adhesive molecules which is described is the integrin, which keeps the basement membrane bound to the endothelial cell lining. Now these are the red cells which are flowing into the capillary lumen, right? So this is a luminal part where the blood cells are flowing. Now the next we see there are small pores. These are small pores which are existing between the endothelial cells. There are small intracellular clefts which are closed by certain gates. Now what happens when you talk about the molecular size? When we talk about transfer across the endothelial cells, so the molecules which are smaller than the pore size, like urea, glucose and ions, can go across these small pores without a problem. They go inside. But there are other ways, but this is one of the mechanisms, and this is called the paracellular pathway. So all molecules which are smaller than the spore size will transgress across, and this is a paracellular pathway because it goes between the cells, the space which is available. Now what happens to water? The water which is there goes through a special channels called aquaporins, and it goes across the cell. It doesn't go between the cells but across the cells, because there are special gated channels which allow water to seep through. Now the molecules which are larger than size of the pore cannot go across through the endothelial cell, and these are protein molecules. Right? So the proteins need to be transported inside the cell by a different mechanism. So what is the mechanism available? The glycocalyx, a part of glycocalyx, has a receptor to which the protein binds. So this glycoprotein or the glycoprotein, which is a part of the glycocalyx, has a receptor to which the protein would bound, which are high molecular weight proteins, and by the process of endocytosis, it gets delivered into the interstitial space. So this is a pathway which is called as the transcellular pathway. So the proteins go across through transcellular pathway described as endocytosis. Water also goes transcellularly because of aquaporine channels which are available. So this is how these molecules are transferred across the cell. Now I want all of you to have the attention, please. Just for 10 minutes, focus with your tired minds, because something which I am going to describe, which is a little difficult to grasp when you read. Now we'll talk about this glycocalyx now. I told you this glycocalyx is a non-circulating portion of the intravascular volume. This entire part is the vascular lumen, all of it. But when you look at this, the vascular lumen, which I have shaded bright red, is the only place where the flow is taking place. Right? Rest of it, the flow is not taking place. So you have the endothelial cell layer here and you have the vascular lumen here. What is between the endothelial cell layer and the vascular lumen? You have the endothelial surface layer. And this endothelial surface layer is what constitutes the endothelial glycocalyx. Now this portion of endothelial glycocalyx is attached to the cells. But there is a portion in the pink part which is not attached to the cell. And if you look at this non-attached cell part of the glycocalyx, this is a soluble form of glycocalyx which is shed away from the attached part. And this leads to the viscosity of the blood, and all the cellular elements like the plasma protein, specifically albumin, is absorbed into this part of the glycocalyx. So it is this soluble portion along with this plasma proteins or the albumin which forms a gel-like structure which is on top of the hairs. Is it clear? So this entire thing is called the endothelial surface layer or glycocalyx layer. One portion is attached, that is the hair, but on top of the hair or the tip of the hair you have a gel-like structure which forms the soluble portion of glycocalyx, which absorbs antithrombin, which absorbs albumin. So all those things are absorbed into this area. So this absorbed proteins and glycocalyx side chains and a part of hyaluron are the composition of this soluble glycocalyx. And this is a vascular lumen which you have all the cellular elements which are flowing through. The RBCs, the WBCs, the platelets. Now you find that all these cellular elements are kept away from the glycocalyx. They are no way in contact with the glycocalyx. They are not allowed to come in contact. Because once they come in contact, then the entire physiology changes. And that is what we will talk about in a little while. So remember, these are some of the glycocalyx which has been shed off from the soluble portion, and whatever is shed off from the foam is in dynamic equilibrium. Even the albumin remains in dynamic equilibrium. But what you need to remember is that this part of the vascular lumen is red cell exclusion zone. There is no red cells here, no white cells here, no platelets. Nothing is present here. Right? And especially in this part of the glycocalyx, there not even the proteins come into this part. It is totally empty. So these side chains which are present on the glycocalyx are all negatively charged. So negatively charged glycocalyx repulses all the plasma proteins out except albumin, because albumin has got an amphoteric structure. So that's why albumin only can bind to these glycocalyx side chains. So this is a sub-glycocalyx space which is totally empty, and it remains empty like this, and this is a base part of the glycocalyx just above the lumen. That's why I have just colored it white. Now let's go to a little more detail. Kindly bear with me, because these are certain important things if you want to understand the function of glycocalyx. It has taken a long time to draw these diagrams and animate them. So the glycocalyx is made up of two structures. One is a proteoglycan, which is a core protein, right? And this proteoglycan is specific because it has got number of side chains. The other part of the glycocalyx is the glycoprotein. Remember in the transfer I told you how the albumin binds to the receptors? So the glycoprotein has the receptors to bind to protein, to bind to cytokines, to bind to platelets, to bind to white cells. So all these things are bound on glycoprotein, which is a part of glycocalyx. And what is proteoglycans? The proteoglycans here are actually having a lot of side chains. And these are side chains called as glycosaminoglycan side chains, or in short we call them as GAG side chains. Now these side chains, I told you, are all negatively charged. So you have dermatan sulfate, you have heparin sulfate, you have chondroitin sulfate. Sulfates means they are all negatively charged. They are sulfate molecules. That's why they keep repulsing all the cellular elements away from glycocalyx so that it doesn't get destroyed. Right? There is only one side chain which is not attached to proteoglycan, and that is hyaluron. So that's a free floating kind of a side chain which intermingles between the side chains and all. Now what does this do? Why do we have proteoglycans? Now proteoglycans are like a tree which has multiple branches. Now what do the branches do? They provide shade under whosoever stands under the branches. You have shade. So this is precisely what the proteoglycans and the side chains are doing. They are protecting the glycoprotein receptors from getting bound to any of the cytokines or any of the cellular elements. So they form a tree, a shade covering the glycoprotein element, so that none of the bad elements combine to these receptors. They keep them away, and the integrity of this endothelial cell membrane is kept intact. Now these are oligosaccharides, just go through it, and these are the thickness of soluble proteoglycans. I told you have antithrombin 3. If all the cellular elements are adsorbed onto the glycocalyx molecule, and these are the albumin which is adsorbed here. All the proteins above 67 kilodaltons, higher plasma proteins, are in the cellular, or goes with the cellular element. They are not attached to the glycoprotein at all. Now where do we find this glycocalyx? This is there throughout the vascular structures. All the vessels have this, but we have different type of capillaries. Now there are three types of capillaries which are described: non-fenestrated, fenestrated, and discontinuous. So we are talking, till now we were talking about non-fenestrated capillaries, which are impermeable ones. But on the other hand of the spectrum, you have discontinuous capillaries. So discontinuous means there is a gap between the basement membrane and the cells. A wide gap is there, and the gap is about 180 nanometers. They have high permeability, and these are the ones which are present in liver, like you have liver sinusoids, the spleen, and the bone marrow. Right? So these are the ones which are present in these areas. And these are basically meant for the phygoendocytic properties. The glycocalyx here, if I've shown you, is not present to that extent. Most of it is destroyed. The destruction happens because it uptakes hyaluronic acid, and this removes the side chains from the glycocalyx. So it is not effective at all in the discontinuous capillaries in liver, spleen, and bone marrow. Now another important aspect: fenestrated capillaries. Just pay your attention now. Fenestrated capillaries have a pore size of about 65 nanometers. But if you look at the podocytes of the individual cells, they reduce the pore size to 6 nanometers. And this is where you have glomerular filtration occurring in the kidneys. So these are relatively highly permeable ones. We have them in exocrine glands, in interstitial mucosa, and renal glomeruli. Now, the practical aspect. When we talk about giving starch, which has now been banned, and mostly all the starch has been taken off from the shelf, especially in the European market. The problem is the starch can go inside here, but it cannot go across the 6 nanometer. It comes to this point and gets entrapped. So what does it do? So any patient having a lower glomerular rate, if you clog these small channels where the starch can go in, it doesn't have continuous glycocalyx, somewhere it is interrupted. right? And this, if this glycocalyx is removed from here, the starch molecule can go through 65 nanometers, but will not be able to cross the 6 nanometer side. It will just be stuck there. So that is why these starches are off, because they are bad for kidneys, can produce renal injuries. Now, all which I had talked about glycocalyx was these capillaries which are non-fenestrated, least permeable to, and they are present in skin, lungs, CNS and muscles. So what's the importance of glycocalyx? What do we talk about intercellular fluid change? Remember, we had Starling's principle, right? The Starling principle, which was told to us, said that there is one force which drives the fluid out of the capillaries, and there is another force which invites or attracts the fluid back into the capillaries. So the one which is driving it out is the capillary hydrostatic pressure inside the capillary lumen, which will try to drive the fluid out of the capillaries, and the osmotic pressure in the interstitial fluid that will also try to suck out whatever is coming out from the capillaries. So these are the two forces which generally drive the fluid out of the capillary into the interstitial. There is another force which attracts or absorbs the fluid inside the capillaries. One is the interstitial fluid pressure, which drives it, the fluid tries to drive it back into the capillary, and of course the plasma osmotic pressure, which tries to attract or suck in the fluid which is there in the capillaries. So this is what Starling said. And when he talked about these forces, he said these forces are balanced. What do you mean by balanced? Now this is the same thing: plasma, endothelium, interstitial fluid. As I told you, the protein goes through endocytosis through the transcellular pathway into the interstitial space. That's why we get a osmotic pressure into the interstitial part, because the protein is going into the interstitial space. If the protein didn't go to the interstitial space, there will be no interstitial oncotic pressure. And the way it goes normally is through the process of endocytosis, which I showed you is transcellular. Now there are small pores. So if the capillary pressure and the osmotic pressure of the interstitium is which will drive the fluid into the interstitium, and the interstitial pressure and the plasma oncotic pressure tries to push back the fluid from the interstitium into the blood lumen, right? So look at into this diagram. So if I expand the capillary from the arteriolar end to the venual end with the capillary in between, you'll find that the fluid first is squeezed out because of the blood pressure, and later at the venous end the fluid re-enters because of osmotic attraction. So this is what Starling had described, which means that if you look at capillary filtration, the capillary pressure falls over from the arteriolar side to the venual side, and there is a area of filtration and area of absorption which is equal to each other. So the loss of plasma is equal to the gain from the plasma. So ultimately there is no net fluid change. But this was an old Starling's hypothesis, which no longer stands, and it has been revised. So what does the revised Starling's hypothesis tell us, and why was it revised? It was revised because of the presence of endothelial glycocalyx, which was recognized later. So let's go to the figure back and see what changes here. Now, as I told you that these are the glycocalyx which govern the small pores, are on top of the small pores. You have a subglycocalyx space which is totally empty. There is nothing coming into that subglycocalyx space. You have intercellular clefts here, and you find that the protein still goes through the transcellular membrane, goes into the interstitium, but some of it will go into the small intercellular clefts. But there is always a gate which doesn't allow it to go into the subglycocalyx space. So it will be just below the cleft, right? And that will form the osmotic gradient generally now. So if you look at this, the capillary pressure and the subglycocalyx pressure instead of the interstitial pressure leads to the fluid which comes out from the capillary into the interstitial space. And when it comes out through a hair kind of a gel foam, it becomes an ultrafiltrate. It's just not the filtrate which comes out, because there is an adsorption which is occurring at the glycocalyx, and the pressure is continuously falling down from the arterial end to the venous end. So it's a plasma ultrafiltrate which comes into the interstitial space. Now the interstitial pressure is much low, as well as the osmotic pressure on that side is also quite low. So there's not much of the fluid which can go in. Actually, nothing goes back into the capillaries. It's only what is filtered out, but there is no venous reabsorption which we had earlier talked about. There is no venous absorption at all, because there is no way that the fluid can actually go into the reverse direction. Now the lymphatics will remove all the fluid which has come into the interstitial space. So what does it mean? It means initially we thought that the plasma has the highest amount of plasma oncotic pressure because the protein content is there. But with the revision, we find that actually the protein content in the interstitial fluid is much higher than what is present in the plasma. So that's why nothing goes back into the plasma. Everything is just coming into the interstitial space, and whatever is coming into the interstitial space is continuously being removed by the lymph and put back into circulation. So if you look at this revised Stirling equation now, what do we get here? We find that from the arterial end to the venous end, the filtration is gradually falling down. It is coming down slowly, and you get the ultrafiltrate here, but if you look it into a graph from the arterial end to the venous end, you'll find that this area of filtration exists throughout the capillary network structure. There is no area for reabsorption. Starling said that there is a constant force, a net force, which is opposing the plasma capillary pressure. But with the revised Starling hypothesis, this force is declining as the filtration falls or as the pressure falls. So it's a declining force, and instead of the interstitial oncotic pressure, it is the subglycocalyx osmotic pressure which determines the amount of fluid which is going to be filtered out from the capillary into the interstitial cells. So to revise it, the basic points which are different. First, intravascular volume. The red one says as a Starling, and the blue which I write down is the revised Starling equation. Starling said that the plasma and cellular elements form the intravascular volume. But now we know that there is a glycocalyx. It has about 2% of the plasma volume. So it is glycocalyx, plasma volume, plus the red cell distribution volume, which forms the intravascular volume. Fluid filtration and lymph. Filtration occurs at arterial end and absorption from the venous end. That is what the Starling said. But the revised hypothesis says it is an ultrafiltration and there is no reabsorption from the venous end. The third one is that Starling said that lymph doesn't play any role because he thought everything is getting absorbed into the venous circulation. But the revised hypothesis plays that the lymph is the major route of return of fluid back into circulation. So all in all, there are three components which has been revised. There is no venous reabsorption. The rate of capillary filtration gradually falls from the arterial to the venous end. It's an ultrafiltrate. And the opposition to capillary filtration is also on a declining force depending upon the subglottic oncotic pressure with the proteins lying there. What's the functional importance of glycocalyx? Last two slides, just bear with me. Like I told you, these are the proteoglycans, which are trees with their branches covering or shading the glycoprotein receptors here, right? So there are adhesion receptors which are protected by the proteoglycans and the side chains. You have different type of receptors. You have selectins and you have immunoglobulins. The selectins are for histamine and thrombin and for interleukins and tumor necrosis factors. They bind to these selectin receptors which are present on the glycoprotein molecule. While as immunoglobulins are basic adhesive molecules, intercellular, that is ICAM. Then you have VCAM, which is vascular cell adhesion molecules, and PCAM is platelet and endo cell adhesion molecules. So this one is for white cells, one is for platelets, and the other ones govern the immunity. So let us see, this is a cytokine trying to come in touch with the receptor, but it is repulsed back because of the presence of negatively charged side chains which are protecting the receptors. So they cannot activate the endothelial cell or destroy it. Now what happens? These are the neutrophils which act through the ICAM molecules. The platelets act through PCAM molecules, and VCAM are seen in cytokines and when specifically when the neutrophils and the inflammation takes place. Now let's go to the next slide. When there is loss of flow, when there is inflammation, and when you give fluids very rapidly, these three conditions, they will disrupt the glycocalyx completely. So the small twigs, because of the storm or because of whatever damage has occurred, are taken off. And this is what leads to exposure of the addition molecules on the glycoproteins to combine with the cytokines or the cellular elements, and it can generate a transcellular mechanism. Am I clear? That's why loss of glycocalyx, because of all these three things, can lead to a lot of changes in microvascular circulation. Loss of flow, there will be thrombosis because there is stagnation. The platelets will come and combine to these molecules here, and that will lead to thrombosis. Arteriosclerosis, these are mediated by macrophages and monocytes, and they also will lead to immunomodulations. Inflammation, there will be disruption of glycocalyx. It will lead to increased permeability, and through the ICAM molecules, because of the neutrophils are coming there producing microinflammation, it will cause edema. So this occurs with bacteremia, polysaccharide, TNFA molecules, diabetes, hyperglycemia, trauma, sepsis. All of them produce inflammation of this area and cause edema. The shear stress, which I mentioned, was with the force with which the fluids are given, at times will shear off the glycoproteins from there. But it has got a protective mechanism. With the shear stress, there is enhanced nitric oxide production from these glycocalyx, which will keep the vessels dilated. Right? So this is one of the protective mechanisms which exists. Now, how does the microinflammation occur? This is a neutrophil which is trying to come in contact with the glycocalyx. As soon as it comes in contact to the glycocalyx, it gets activated because it has attached itself to the receptor, and then it firmly binds to those endothelial cells, and through transmigration it goes into the interstitial cell, which it should never be allowed to go. And when it goes there, there will be injury to the plasma membrane, the membrane is disrupted, and the cellular swelling will happen. So this is how the cellular swellings and inflammation will cause problems into the patients. So just to summarize, thank you for bearing with me. Remember, glycocalyx has important structures. The proteoglycans, which have side chains, which are tree, which form shades protecting the receptors. Glycoproteins are the ones which have the receptors. You have side chains which are present onto the proteoglycan. The revised Starling equation, the interstitial oncotic pressure is replaced by the glycocalyx osmotic pressure. There is no venous absorption. Rate of capillary filtration increases with the rise in oncotic pressure. Opposition to the capillary force is declining. And these are the factors which can lead to damage to glycocalyx. Thank you very much.